right, so I'm going to go ahead and stop my screen share for a moment here so we can give Gabriella a bit of a better intro, talk about why she's here, how she got became a part of Fundus, and anything else you'd like to share about yourself.
Yeah, thanks so much for producing these kinds of podcasts and these educative things, Emma, and Roland. And I'm so happy to be here and talk about fungi, how to get involved in fungal community science. Um, those projects and initiatives you were just mentioning, Emma, seem fantastic. The SciStarter mission, and also how you defined citizen science, I loved. It was such a just an essential definition you have of a collaboration between scientists and people who care, people who want to make a difference. And so that's really what fungal diversity grew from. I'll take a step back though, and I'll introduce a little bit about myself. I'm also I'm pretty much better from a cold, so I apologize if I'm a little nasally, but I will try to dictate very clearly. Yes, I'm from Utah. I grew up there, and I think I got into fungi because they were really an underground. Um, I was interested in the cultural phenomena of them. Like, why do people have really large reactions when it comes to fungi? They can, people despise them, they hate eating them, they're grossed out by them, or they have strong aversions, or they have these really strong curiosities and passions, and they love them. And what makes these beings so special? And then as I was forming that curiosity, I moved to Washington State and went to college in Seattle. And lo and behold, there's mushrooms everywhere there. And so I think the first really big fruiting or flush of mushrooms that caught my attention were Amanita muscaria, which is like basically the tried and true like emblematic fungi. Then the mushroom, the emoji mushroom, the toadstool, the red cap with white warts. There's actually one right there that has
Oh,
the white spots or warts on the cap, and then this white stipe or stem, but with mushrooms we call it stipe. And they were growing at the base of my dorm, just right in front of my dorm. And it was like the first week I was there, and they were like each cap was this big. I mean, they were multiple for the listeners. There, each cap was like the size of the width of my hand across. They were huge mushrooms, and I had never in my life seen anything like this. I had only seen some field mushrooms, some white field mushrooms growing in my lawn growing up as a kid. And I thought, just what on earth are these mushrooms? Oh my goodness, they were so beautiful. And I was able to, in my case, I was able to focus this passion that I started exploring Washington State looking for mushrooms into my academic focus. I was at college getting a bachelor's, and I was studying environmental studies, and my professors were asking me to choose a focus, and they were like, why don't you just choose the mushrooms that you're studying? And I was like, but how? What does that mean? They're like, I don't know, you tell us. And so it was a very creative. I appreciate some of the guidance that I had with them telling me, yeah, you can focus on this passion. And that also leads me to another topic that community scientists and people who love fungi are so important, especially right now, because there is so much there is so much under resourcing happening for fungi at large, and that has always happened, really, especially in North America for hundreds of years. There's fungi in our culture, especially as it relates to like indigenous people, have used fungi in our culture here in North America. And then as we've capitalized and grown, more infrastructure with this culture, we live in the United States of America, and we have funds and sources and science and people learning things. And fungi have been left out of most of that infrastructure, unfortunately. And so it was a big step for my professors to say, hey, why don't you study fungi? But they didn't know about fungi. They weren't trained about fungi. I had ecology professors and biology professors, and they didn't hardly know anything about fungi. Um, so it was a self-taught thing in many ways, and I would take concepts that I learned in books or online and apply it to their ecological certain inquiries or things that we were learning in class, papers, projects, and they were like, sure, sounds good. And then I graduated college and found out that, well, my senior capstone in college was looking at the knowledge creation and transferal of mycoremediation. And mycoremediation is filtering contaminants, is using fungi, to filter contaminants from the environment. So it's an applied mycology approach. It's looking at this mycelium, or the fungi that exist in the ground, and the fact that they can actually hyperaccumulate heavy metals, toxins, they filter contaminants, they can be really have a lot of potential for land restoration. But I was looking at these two sources of knowledge and saying a lot of the work, a lot of the knowledge is coming from academia, coming from academics, but a lot of the work and the knowledge is also coming from what was deemed as grassroots movements or people outside of academia. And I was really just saying that there's these two places. Why can't they communicate better? And why can't it be more of a closed loop between the two places of knowledge creation? I graduate college and I start, eventually a few years down the road, I get involved with the Fungal Diversity Survey, which is a nonprofit that we're focusing on now, FUNDIS for short. And I realized that what I was studying as a senior in college was really community science or citizen science. And we didn't have that title. We didn't call it that. And it existed at the time, it was 10 years ago, but it hadn't gotten to my spheres yet, the idea of citizen science or community science. So I was like, wow, this has been a concept that I've actually been interested in for a while. And that's really important to me because a lot of people, anybody can have an impact on science and conservation and how we better understand the planet. And we don't have to necessarily be working in academics or be paid for, have an academic institutional seat. We don't have to be at a university or so on to make a profound impact with our understanding of science. And so Fungal Diversity Survey, I'll fast forward now, is this nonprofit I've been involved in for about 4 years. We began in 2017, and we exist to help understand fungal biodiversity and to protect fungi. And so a large part of our mission at the beginning, in the first few years, was really expanding this concept of community science. And wow, there are so many people who are already going out into the field looking for mushrooms, taking pictures of them. And wow, if they just had a little bit of some guidelines to say, hey, you doing that just out of your love, and if you just add your observations to iNaturalist as an example, or if you add them to our fundus programs or projects, it can start to have a science or conservation impact. And so the goals of our nonprofit are to protect fungi in their habitats, to create pathways for people to participate in this, so elevate the voices and the actions of community science, of community scientists, and to increase the public appreciation of fungi. So I'm happy to kind of like maybe let things pause there if you'd like to continue, Emma, with anything else, or I can just very much continue.
No, thank you so much for expanding on all of that. That was really interesting information, and I'm glad to know like your actual story, because I feel like a lot of people can probably relate to that, of being interested in mushrooms or absolutely hating them. I myself hated mushrooms at all ends except for on pizzas for some reason. I love mushrooms now, but was not a fan as a kid. And so it's very relatable to see maybe people who are interested in them and don't really see pathways, because I've heard very similar stories of like there's not really as much research done, or like the research that is done, it's not all over, and it's a tougher one to know about unless you know where to go. So I appreciate you bringing that context in there as well. And apologies, I actually neglected to introduce the last person on the call whose face is visible. So I'm so sorry. I'm just going to take a moment to say, hi, Bob. I'm so sorry. I'm the worst. Good to see you. Would you like to say who you are?
Oh yeah, I'm Bob Hirschon. I do the podcast for SciStarter and the newsletter and lots of other stuff. But yes, and I'm here partly because I'm interested in fungi, and it's fungi, right? Not fungi or fungi. You say fungi.
I was going to say, you can say however you want. The
Tomato,
important
tomato.
that you're saying it. teach this immersive weekend that just happened this past weekend in October, where we have 4 instructors, 4 of us mycologists, and every single one of us does it differently.
Oh,
Um,
okay.
there's
All right.
I chose fungi. I think it's a prettier way of saying it.
And we say fungus too. We don't say fungus. So
Exactly. And
fungus goes with fungi. Yeah.
right. And my Italian, like Latin background,
There we go.
fungi, we've just taken off the H. So I don't know.
That's it. Okay. Well, that's the correct way. We just have decided. And I just had a question because they're really cool, obviously, mushrooms, but um, what's their ecological not that everything has to be important for reasons other than it's just cool to want to find out more about them and protect them. But could you talk a little bit about why they're so important ecologically?
Absolutely, Bob. And I'm sorry that we had a little bit of a crossing wires at the beginning of this episode, but I just want to thank you as well for being here, and thanks for doing what
Oh,
you do.
sure. And you too.
Yeah. Well, thank you. I thought you'd never ask. Mushrooms are so beautiful, and they have so many, like, people love looking at them. But of course, the reasons why we want, as our nonprofit, for people especially to go out and help us understand where they are and who they are, and what they're doing is, well, I'll start off by saying that we only have an understanding of 5% of that. There's an estimated 2 to 10 million species of fungi that could exist on the planet, and we have scientifically described 5%, about 155,000 of those species.
Wow.
And when we look at maybe plants as a comparison, we've probably described about 90% of them. So it's the inverse, right? We have only described maybe 5% of fungi, whereas 90% of plants. The same thing goes with vertebrates. With vertebrate mammals, we've maybe described 90% of them. And so it's important to help understand science, and to help we don't know the distribution patterns. eBird is a really great example of a successful community science model. They help they're able to map like bird migrations, and say overnight, tomorrow night, like this whole species of bird will move into the Gulf of Mexico, maybe, or up the southeast. Whereas we can't say that for mushrooms yet. We don't have that much data, and we really need the people to do that to help us create that data and help create the understanding. And why are fungi important? We would not have life on our planet without fungi doing what they're doing. Our planet would not be habitable. It would not be functioning the way that it's functioning right now. We would not have soil creation. Soil is we wouldn't have food, we wouldn't have agriculture, because fungi make this thing called glomalin, which actually is this basis of soil. Humans can't make glomalin. We wouldn't have the processes of our planetary resiliency. We wouldn't have decomposition. If it wasn't for fungi, we'd be walking around with just all the waste up to here, but actually it's compacted and eaten by fungi all the time. Bacteria and fungi, but fungi are essential for this. We wouldn't have land connectivity. Plants and animals rely on fungi, this forgotten kingdom, or kindom, which I like to say because it just becomes true of our connectivity as well as gender neutral. And so the plants actually rely on fungi. 90% of plants connect to the mycelium, this underground part of fungus, right? Not all fungi produce mushrooms. Sometimes they do, and when seasonality is right, a certain fungus might produce a mushroom in the fall time when rains come. 90% of plants need their fungal hosts. The plant and the fungus participates in this symbiosis where the tree can actually receive nutrients like carbon and phosphorus, and this exchange happens where the fungus receives sugars and fats from the tree. And so it's this beneficial symbiosis that's making it possible for at least 90% of our plants on the planet to actually live. And the same, it's such a foundational web of life, these fungi in the mycelium that allow bugs and different trophic levels, without the mushrooms and without the mycelium, we wouldn't have the stability of our animal kingdom as well.
Yeah. I was going
Yeah.
to say, and if we hunt them, are we hurting that diversity?
Yes, thank you for that question. So the mycelium, as I said, permeates the land, and it exists 365 days around the year. It permeates like barren deserts, the bottom of the ocean. They live in places where you would least expect life to thrive. And when we pluck a mushroom from the mycelium, we're not actually injuring the whole organism. It's very similar to plucking an apple from a tree, but the mushroom is very important for sporulating and for being able to spread its spores, and because the spores oftentimes do come from the mushrooms. And then when you take the mushroom, you're reducing the chances of that fungus to be able to replicate and spread its spores and to continue, right? Because once spores fall, they have another place to land and the fungus can continue to exist. So I want to say that just with any foraging, just with anything out in nature, right? We're not asking you to go and collect every single mushroom you see there right now, right? It's like, as respectful, ethical humans, we don't take 100% of the mushrooms that we see. If we're taking mushrooms for science, we want to know why, right? Why are we taking the mushrooms? Okay, if it's for food or science or medicine, it's generally a really established practice that we only take 30 to 50% of the mushroom body of the mushrooms that are present. So if you have this really big, beautiful maitake that's out there, or this edible mushroom that you're finding, like a chicken of the woods or something, just take half of it, because not only does it allow the mushroom to sporulate and continue to thrive, because we want that to happen, or else you come back the next year and the chicken of the woods might not actually be there. So you want it to
divide them? Oh, okay.
Yeah, they can continue every year that way, but also you're leaving mushrooms. Exactly. This is a beautiful example here.
Wow.
That's actually not a maitake. That looks like an armillarius, but that's a honey mushroom. So it's funny that you don't believe what the internet tells you about mushrooms. It's important, especially for edibility reasons. That first one, those are maitake. Yeah, so you can take, a part of it. And if you're taking something for science specifically, that's a great reason to go out and pick mushrooms and come back home and dry them and save specimens. Like, I know that viewers can't see this, but some of these specimens in this bag I have that have been dried. And the reason why we are asking for specimens, which I'm kind of going pretty ahead here. So how we can ask community scientists to help us comes in a variety of ways. You can really go like all the way. You can go in and go out and actually bring home specimens, mushrooms, dry them. We have all the information on our website about what to do, how to take high quality information. We have field data slips where it gives you a photo checklist. Are you taking pictures of the top, the underside? What's the spore color? What's the habitat of the mushrooms? What is it growing on? Is it growing on wood, soil, grass? You can go all the way and actually take specimens that you can then send to us for DNA sequencing, because part of understanding a mushroom identity actually has to do with DNA sequencing, believe it or not, because there's so much opportunity, there's so much discovery to make, and a lot of the mushrooms we can't thoroughly know them without looking at their genetics, unfortunately. They're just so they have so much variety, and we're seeing more and more that there could be two species that look pretty much the same to us humans, but in fact they have different genetics, and that could give us different information about what these species are doing. It comes down to the genetics in order for us to look at, okay, if mushroom A has a genetic different genes than mushroom B, mushroom A has a different genetic expression and is probably doing something differently in the environment than mushroom B. So DNA sequencing can really help provide a valid ID to help us understand the fungi that are out there. That's really the main question that community scientists can help us understand, is what fungi actually exist. First off, the easiest thing to do to get involved is to take pictures of the mushrooms that you're finding. High quality photos go a really long way. We have a lot of those educational materials online. We have videos, we have it written out. Taking a photo goes a long way. And we don't want a photo of a mushroom from like, just the cap of a mushroom from 10 feet away. That's not going to help us look at the mushroom. And it's okay, because a lot of us haven't learned in school or growing up what parts of a mushroom are. We've learned about trees and roots and leaves, and that comes more easily to us. But there's a lot of catch up to do. So to take a good picture of a mushroom, Tay right here, Tay Bright is one of the people who work with Fundis. And Tay, if you are viewing this discussion, she's taking really great pictures of the mushrooms. So you want to pick up a mushroom entirely, its full base, including like the very bottom of the mushroom that might be hidden, might be still underneath the grass or in the moss. And if you don't get the base of the mushroom, it actually becomes really hard to tell what that mushroom is. There's a lot of mushrooms that have unique traits on the very bottom of the stipe. Yeah, there you go. So that's a great picture where you can actually see there's some like oranging. The bottom of that stipe there has an orange discoloration, and it's an equal distance of a stipe, whereas some of the stipes have these big like bags or vulvas, or they have different shapes. And so it just becomes like, learning about what potentially to look for with mushrooms. And so it's just a really great practice to be able to flip your mushrooms over, take a picture of what's on the bottom side as well as on the top side, and then put your pictures on to our lovely partner that everybody loves so much, iNaturalist, who does fantastic work. And iNaturalist, on iNaturalist, we actually have particular projects that we'd love for our mycofiles to add your observations to. So if you're putting your mushroom pictures on iNaturalist, we have an overarching project called the Fungal Diversity Database. The Fungal Diversity Database, and this is the place where we want all of our high quality observations to live, because we can collect them this way. It's a vetted database, so we have volunteers who are helping the participants, helping the people who are adding their observations. Yeah, this is the Fungal Diversity Database. So we have, it looks like over 217,000 observations right now, made by 1,700 people, and over 6,000 species so far have been documented. So this is across North America. It goes to Central America, Puerto Rico, and up to Alaska, Canada, even Greenland we've included. And we really want, this is the easiest initiative to ask of our community scientists, to add your mushroom pictures, your mushroom observations to this project. Once the observations land here, it becomes a vetted database to be able to provide to land conservationists or landowners or scientists. Whereas the mushroom observations, if they just live on iNaturalist, they become more difficult to find, and they become less trustworthy if it's not in a database like ours. And they kind of don't have as much impact or as much scientific value as they do when they're collected together.
I'm gonna pause you for a second right there. I'm glad that you mentioned, I'm glad we were evolving into like how to actually get involved. So this is great. This is going really great. So I'm curious then on iNaturalist for this particular project. Did I hear you right in saying that it's already vetted before it gets added here? So it has to be research grade before it's on here, or how does that work?
Research grade is a funny thing with mushrooms on iNaturalist. So the vetting thing that we talk about just means that we have volunteers who are going through, they're thumbing through the observations that do get added to the project. And periodically they might remove an observation because it might not meet our quality standards. And if they do, they will just comment on it or message you and say, hey, the reason why we removed this is because we couldn't really see the underside of the mushroom. We couldn't tell if there were gills or pores or teeth. And in that case, it's just not a high quality enough observation. So we've removed it, but your other 10 observations are awesome. So keep doing the good work, and thank you for doing that. And so we have that degree of volunteer engagement, but we also have volunteer identifiers who are trustworthy sources for scientific ID. So this is, we just have people like John Plischke, Sigrid Jacob, who you can see here actually on the project themselves, Mandy Hackney, Mandy Mushiai. They're helping give IDs more accurately than more accurately and more quickly on our project. So that's a plus for if you're a community scientist. Why would you add your observations? Because you can get an ID that is more accurate, and you can get it quicker if you add it to our project.
That's great. I haven't, I know that iNaturalist does a lot of work to do the whole switch from casual observation to growing up to research grade, but it's good to know that we make mistakes still. Like we have oftentimes, like in City Nature Challenge, for example, that one weekend with the ID extravaganza afterwards, where we're going through an IDing. Sometimes it's so hard. So for the City Nature Challenge that I'm a part of, we enlist the help of the people who are most commonly confirming these species for specific species of plants and animals that happen in the City Nature Challenge that I'm a part of. So it's good to know you guys have that built in into the system to make sure it's accurate from the get-go.
Exactly. And that it's interesting with fungi because there's so much discovery. There's so many new species. There's about almost 2,000 species of fungi that are being discovered every year. And on iNaturalist in particular, the research grade doesn't fit nicely with fungi right now. Fungi don't like to be boxed in, and they will teach us that in any way they can. And so a lot of the species, the new species that we're finding, like there's just species that are floating out there that are like psilocybe CaO2. Like nobody's actually sat down and written out has made a species name for some of these species. So a lot of them don't actually exist in iNaturalist yet. So that's so a research grade observation for iNaturalist, like the project you had pulled up, it said that we have over 6,000 species documented. That it's very hard for that to actually be a very true number, or at least, yeah, we have 6,200 species, because a lot of the species they don't have a field in iNaturalist yet, or they don't have actually a description yet. So they don't have a name yet. So a lot of the commons mushrooms, sure, but then there's like more the least common, the newer discoveries. Like if you other projects you can look at for this kind of stuff, especially is we have other work that we're doing, and some of our work is actually staffed. And our CA Fundis project is I can bring this in right now. CA Fundis, we're being funded by the state of California and the California Institute for Biodiversity to help describe to create an inventory of fungi in the state. And so we're using iNaturalist, and we put our DNA it's called CA Fundis Year 1 or CA Fundis Year 2 are the projects you can look up on iNaturalist, and you can see real time what we've been finding to help understand the diversity of fungi in the state. But a lot of the species are quote unquote like, unconfirmed or they're contested on iNaturalist still, because we have the DNA sequence, and we have our people working on it saying we're going to call it this, but we have no perfect fit for the iNaturalist taxonomical library that it fits in. That's a more nitty-gritty thing.
And so for us beginners out there trying to contribute, how do you know something is a fungus? Because, they have the things with the caps and the stems, well, that's a mushroom, but there's so many things that are like tiny, almost microscopic. There's like blobs, there's things like, all sorts of weird things. How do we know it's something that we should report?
That's a great question. And that's why that mushroom education is so important. So we have a lot of resources online. Your local mushroom club, if you live, I think Emma had said that they're around the Washington, D.C. area. There's a great Washington, D.C. club. I'm in upstate New York. There are plenty of mushroom clubs. Those are fantastic places to get involved. If you really just need to make, like, I don't understand what the difference is between a mushroom and a plant, go to your local mushroom club. The North American Mycological Association, or NAMA, they're the hub, they're the mothership of most of the clubs. So if you go to their website, they'll list out all the clubs, and you can find ones that might be in your state, or you can use the internet. That's a great resource to help go out with people, or just go to a few talks, start to read, get a list of books. But yeah, you're right. A lot of mushrooms don't look like caps and stems, and I was just holding one that looks like a star, and a lot of mushrooms even they can you can start looking for different mushrooms, especially relating to the habitat that they're growing in. A lot of mushrooms only like growing on moss, or some mushrooms even parasitize other mushrooms or parasitize insects. And like there might be some tiny little weird club growing out of a spider, and you have no idea that what you're looking at is actually a mushroom. But I would say that that's a great reason to just have a curious mind, and to take photos of nature and put it on iNaturalist anyway, and
How
help
about
understand.
truffles? Do we have truffles? Because they're underground, right? How do you find truffles?
Yeah, truffles are.
I'm getting carried away here.
No, you're fine. You're fine. Truffles are, yes, truffles. There's different kinds of mushrooms that don't fruit above the surface. There's a lot of mushrooms that fruit in the ground, and those are called hypogeias mushrooms. When you're looking for I mean, sometimes you can see them like sticking out of the ground, like as a rock does, and it's just kind of the trained eye. You know, if you're looking for mushrooms, slow down. That's the biggest tip. A lot of people are going through forests and they're looking at the trees and looking at the birds, and you'll find more mushrooms going just crawling. I joke that it's like much more of a mushroom crawl, or like you go very slowly when you look for mushrooms. So if you slow down, start to notice your surroundings, your immediate surroundings, look down, look at the trees, because mushrooms grow on trees as well. And use your smell. A lot of the times you can start to smell mushrooms before you even see them, which is fascinating.
Wow.
And then if you're really looking for these truffle mushrooms, follow the squirrels, and see if the squirrels, like, just sit in a place, see where the squirrels are going. And if the squirrels are stuffing a lot of things in these holes in the ground, there's a good chance that there are truffles growing in that area. And I do want to say though that, you know, truffles, ethically, you know, we should be not removing too much leaf litter or not digging big holes in the grounds and things like that. So if you're really interested in truffles, I would recommend going to your mushroom club and finding somebody who has experience looking for truffles, and they'll start to show you different ways that you can look for truffles in a way that isn't too super destructive on the environment. And a lot of the truffles, though, there is lots of discovery to be made with truffles.
Okay. Now, and so how do we contribute other than taking pictures?
That's where I was headed to, actually. Let me, before you get started, I want to show one slide, since this is one method of getting involved that's listed on the SciStarter project page for Fundis. And it seems like there might be quite a few other ways, including like sending specimens and such. So we'd love to hear about those too. But for anyone who is thinking about the first step, since we know about different ways in which to walk through a forest or crawl through areas to find mushrooms now, if you want to start contributing observations, these are the steps to do so. So iNaturalist is a SciStarter affiliate, which means if you use your username from iNaturalist and add it to your SciStarter account info and settings, then your contributions will be counted over time. So you can see just how many observations and how often you observe, et cetera. And you can find that on your dashboard. So step one. Step two, the project page for Fundis is right here, looks like this. And in the steps for, which I'll drop back, in the steps for under description, there's a link to go straight to my best find on the website. When I saw that, I was like, oh, this is perfect. It goes through the exact steps of signing up, creating observations, some advice for iNaturalist, the project on iNaturalist, and then also the minimum quality standards checklist, which you were touching on a while back ago. So I was very glad to see this hang out there, and even a video to watch 1 minute of how to photograph a mushroom. So you have a starting point for all those things. And I'll go back here and you just start observing. Now, beyond that though, what else is involved and how else can people assist? Or any additions you want to make to that too as well?
Absolutely, yeah. So, right, that's the easiest thing to do. Take pictures of the mushrooms, put them onto iNat, tag Fungal Diversity Database, add those observations to the project, Fungal Diversity Database. I'd say the more involved processes start to be looking for rare or threatened mushrooms, you know, certain target mushrooms. That's probably the next step. And so we have a project called Rare Fungi Challenges. And the Rare Fungi Challenges, so far we have 3 of them. They are these challenges that are for a particular region. So this one on the screen for viewers is the Southeast Rare Fungi Challenge. We also have the Northeast of North America and the West Coast. And so we've put together a list of 20 target mushroom species that we think, and we've worked with expert mycologists and conservationists to create these lists that we think are rare or threatened. And we want you to help us find them. So a lot of the mushrooms don't have a threat status because we don't have a good understanding of how many there are or where they even exist. And the Southeast Rare Fungi Challenge is the newest challenge. Some of the mushrooms in here, it's been running for a couple of years, but I will say the challenges that are a little bit older, like the West Coast Rare Fungi Challenge, began in 2020. So we now have 4 years of data at this point, and we've been able to see that. For instance, one of the mushrooms, Bondarzewia berkeley, has been found maybe 100 or 200 times. And then another mushroom, a purple Romeria, purple coral, hasn't been found still yet once. And so it's really helping us understand, okay, based off of actually promoting these species for the first time, what mushrooms are actually rare? Just how rare are they? We're really understanding this, and this is important. Community scientists can help us so much in this way. So you can go on our website, print off these little pamphlets that we've made. They'll tell you where the mushrooms we know of, where they might be fruiting, what states they might fruit in, what habitats. Maybe it's a swamp or a wetland or like a pine forest or something. So if you're going out and hiking, take one of the pamphlets with you and be looking for the mushrooms. And if you happen to find something, take a picture of it, and it just might be one of the rare fungi. I'd say the more sort of intensive way for people to get involved are to become involved with our rare fungi, our Fundis local projects. So we have a network of local projects around North America. We've had over 200 register with us. Anybody can make a project, anybody can join one. Each project is a research study to look at fungal biodiversity in a given region. And so academics, professors, educators, parents, kids, students, they can make or start one of these projects, and it can be as simple as the fungi in your backyard. Great, I'm going to make a project of Gabriella's mushrooms in my backyard, and I'm going to go through my backyard once a week, every single year, and observe the mushrooms that are fruiting. That is probably the most helpful thing you can do as a community scientist, is just go through an adopted piece of land for months, for years, for a lifetime, and help us understand the mushrooms that are fruiting on that piece of land. So what viewers are looking at is part of our umbrella project on INET. So the umbrella project includes a lot of these individual projects. And so as you scroll down, you can see that there's the fungi of Pennsylvania, plus there's neotropical fungi, there's Alabama fungi, there's fungi in Guam, central New York, Northwest Georgia. People can adopt a whole state, a park, and that is so fun because you have this rhythm. Maybe it's every 2 weeks. Maybe you go out and it's an open call. Anybody in the community, come with me. We're going on the, I started the northern Utah funga. So funga is like flora and fauna. We ask everybody, we are partners with Fungi Foundation and Juliana Furci, who's really trying to get the third F included into flora and fauna. So whenever you hear that, just add the third F, which is funga, which just means the fungi that exists in a certain place or a time. And Northern Utah funga, well, I've integrated that project with the Mushroom Club of Utah. I don't live there anymore. I live in New York, and they're still doing it. They're still going out, adding their observations to the project, helping us understand the biodiversity of the place.
How about our listeners and viewers in Europe and Australia and Asia and around the world? Are there corresponding things that they can join?
That is a great question as well. I would say there's one of the nonprofits that does a lot of beautiful work worldwide. Well, two of them. One of them is the Fungi Foundation, as I just referenced, and they have a branch called Fungi Education that does a lot of worldwide education and provides a lot of resources, learning resources, particularly for people who want to teach about fungi and they're trying to get fungi integrated into public school curriculum worldwide. But they have a lot of resources online. They're doing other forms of study that you can look into if you're in a different place that's not North America. Also, SPUN, the Society for Protection of Underground Networks, is doing work around the globe, and they are doing a lot of soil-based mycorrhizal work. So they're doing like soil samples to try to map the fungal biodiversity across the globe, and they might be looking at extending into community science. But they do a lot of you can apply to them. Anybody can apply to them to do work with them, but they might try to reach out into community science spheres a little bit more. And then I would say, just if you like what we're doing as Fundis, just start doing it wherever you are. You know, we can't do everything across the globe. We've already had to choose to focus on the continent, which is already so much. And so if you like what we're doing, just reach out to us and say, hey, I love your project, what you're doing. You know, the rare fungi challenges, or the projects that we live in Nepal. Great. You please use this structure if it's useful for you, and start hosting your own work in Nepal and start building community science that way. And we're happy to help.
I have a really quick one. Is Seek useful for this, the Seek by iNaturalist? Because I've used it and it seems really quickly to identify things. I don't know how accurate it is, but it seemed really helpful.
Yeah, Seek. I really encourage iNaturalist, because as a community scientist, your observation doesn't live openly if you use Seek. If you use Seek, it's important for just myself if I'm using Seek, and I can look at that observation again. But if I'm using iNaturalist, my observation lives on the internet, and then scientists and conservationists can see Gabriella found this mushroom, this psilocybe, which happened in California. We found some new psilocybin, new psilocybe species in California from our work. Other researchers, other people can see that and say, oh my goodness, you have a specimen that I can then receive and be shipped to Europe to do DNA work or phylogenetic work, or it becomes this interface. It's a very important tool for community science to have this like see-through interface, where like scientists can use it, community scientists can use it, and the data can be accessible to everybody. So Seek is an iNat like partner. It's made by iNaturalist. I don't know if their AI can recognize mushrooms as well as iNaturalist can. I haven't done the testing myself, but I would say that iNat all the way, just because you can actually, if you make the second, just press post instead of press exit on the app, your data can actually become so useful and so helpful for science at large.
Okay, cool.
To add to that, Bob, for Seek, sometimes it is tough for it to identify because it doesn't give you the what do I see like option that iNaturalist does. So if you submit an observation on iNaturalist, you get to go through the list of options for a species, and Seek doesn't necessarily do that. They just try to tell you exactly what it is, and so it's not always as accurate. So in this case, especially when there's like an issue of the species doesn't exist yet in our taxonomic levels of the kingdoms and whatnot, all those. That makes me think iNaturalist would be better for that reason too, just because if we don't know, we can get as close as possible and then hand it off to the experts on the fundus side. Get the different options. Now, since considering time, I just want to check in and ask about outcomes. We've talked a lot about like what the purpose is and what we're trying to learn, but I wanted to bring us back to something we were talking about just before we started this this call, to see like what's next for Fundus. What are we actually what are the outcomes so far? Anything you can tell us about that, what we should be excited for being able to know now too.
Yeah, our community science, we are going into a wave of re-engagement in the next year. So we'd really like to build up those programs and the support that way. So look forward to more assistance with community science. We'd like to fill out the map with the rare fungi challenges and really like bolster our local projects and so on. And our work with CA Fundus has been incredibly inspiring. It's the first statewide, it's the first state to include fungi in their inventory in this same in this scale. And so this has been fascinating and super exciting. We've been working with so far a 17% new sequence rate, which means that out of the, we've probably collected 9,000 collections so far in California of mushrooms. And out of the ones that have been DNA sequenced, which have been so far maybe 5,000 or so, we're working with 17% of them don't have a matching DNA sequence, which means that they're either unsequenced historical specimens, so specimens that are living in a fungarium somewhere that haven't been DNA sequenced yet, or they're totally new species that nobody's documented yet. So there's a lot of data that we're starting to find and create in California. And, you know, really as a conservation nonprofit, we use data. Data is such a tool to be able to assess the status of fungi and how to help them. So we've made this graphic last year with Sam Bruchet, who's a designer with, does a lot of design work, and he's also helped us. And, you know, a lot of people think, what is fungal conservation? And how do we protect fungi? They might even think that, you know, a rare fungus, that's a new concept. I remember when that was a new concept to me, that, wow, of course, mushrooms can be rare. You know, everything has its habitats that are slowly declining, and that especially with mushrooms who rely so closely on the trees or a particular niche, they might not be thriving at this point when we have changing climates. So we're really excited to be like getting the wheels of this cycle rotating. The common understanding of how to conduct fungal conservation is the same thing. It's assessing the needs. What's the status of fungi, of fungal biodiversity? Then you make plans to help, you know, whether it's okay, we're prioritizing the needs. We have engagement strategies. We have community science. We have particular goals, and then we act them out. We enact them, and then we assess how that went. Plan again. Act. It's like, you know, how you form projects in general, but it can be very elusive when there's a new concept of conservation going. Like, how do you conserve something? Well, let's look at our needs. Let's look at the priorities. And so with California, we've had a couple of years of building data now from both community scientists and our staff who are skilled people who are actually paid to go out and collect mushrooms. And we're getting to look at, okay, based off of our data, based off of what we've seen, how can we make perhaps like a California fungal conservation campaign? What would our initiative be? So we're starting to rev up our engines into being able to at least say something for a particular state now that we've seen so much data from California. And also in the volunteer places where we work, like the rare fungi challenges, which I want to say too, that anybody can join a rare fungi challenge too and volunteer internally with Fundis. We have lots of volunteer opportunities, which is a great way to get another step up in involvement. But the data we're building in those places, we want to help perform IUCN Red List assessments. You know, the data that we're actually building, the species that we've chosen, a lot of them are data deficient. We're the ones trying to get community scientists to help us build the data, and every year or so we're trying to see, okay, do we have enough data to actually put them on the red list yet, or to change something about the red list? And so we'd like to have we're gauging our conservation strategies currently, and also how to really uplift our community scientists.
Excellent. That's
Nice.
awesome.
Okay. Let me think.
Did you have or let me switch back to that. It's kind of incredible to think that there isn't enough data, because I feel like we see mushrooms all the time, and so it ends up being like, how do we not have enough data? But we don't half the time. There was, not to relate it to another outside of funga and move on to fauna briefly, but there was a research project recently where they looked at iNaturalist and a couple other, I think it was eBird and a couple others, to determine what were under or data deficient bird species, and they were able to identify 126 species of birds that have not been looked at. They're lost to science because they haven't been observed in 10 years. And so I would love to be able to see that in the future for fungi too, to be able to look at if we can determine that off of all of these strategies of being out there and building the data, where we might be able to find that out too, just from looking at the data instead of and so those rare fungi challenges could be through just looking at the data once there's enough out there. I don't know if that's going to be possible, but that would be cool. That'd be
I
great.
would love to know more, Emma, if you can send me what that's from. I would love to know what that is, because I'm thinking like, well, I mean, there's been tons of mushrooms that people haven't seen since like the 80s or the 90s or so on.
The Lost Mushroom Project.
I'm dropping it in the chat just in case anyone else wants to know about it too, but it's this is a report from Science Friday, but it leads you to the actual research project too. I think they're called the Search for Birds.
Are there any state fungi? You know, like right now there's a big thing in DC. They're trying to have a state amphibian, the red-backed salamander. And so if every state, you know, kids, mostly kids, it could be adults too, vote on the state fungus, that would be a really cool thing. And then that would get people, oh, fungus, what, you know, and they'd start looking, what is that fungus? So maybe that would help. Exactly.
That's great. The amphibian, the amphibian files are getting a state amphibian. There is a movement for that with mushrooms. I believe there's maybe 5 or 6 states only with one so far.
Well, that's pretty good. I didn't realize there were any. Okay.
Yeah, and we were I was a part of the movement getting the porcini for the state fundus in Utah, and that happened a couple of years ago. There's a few of the states have some like burners in that with that directive, but the more that we can do that, the more presentation. Exactly.
Yeah, but Pennsylvania has one there, the mushroom capital of the world in Kennett Square. It's where all the mushrooms come from. Where I'm just visiting there next week, so that's why I know that.
Pennsylvania has a state mushroom?
Well, they must. I suspect they do, because they have the mushroom. Kennett Square in Pennsylvania is the mushroom capital of the world, where half of the mushrooms in the United States come from that one town. And a high percentage of mushrooms in the world come from this one little space in Pennsylvania.
Yeah, they don't, to my knowledge, Pennsylvania, which Pennsylvania would be there's a lot of mycologist friends too in Pennsylvania. I'm sure that could be
mushrooms there.
Yeah, it's interesting they're not on here yet. Maybe they just have too many options and they're like, we don't know.
They would fight because there's so many. It's an industry there. So no, our mushroom should be our mushroom. Yeah. So
yeah, there's only a few. You can see you're bringing them up. Yeah, Utah has the porcini. Texas has this beautiful earth star, the Corioasis geister, and there's chanterelles. There's
wow,
Vermont.
so it really is happening. Oh, Massachusetts.
Oh, yeah, some of them.
Puffballs, those puffball things.
There's a few proposed. I live in New York now, and so we're trying to help here with the Lactarius peckia. And you can go into, right, it becomes a statement. It becomes like, okay, Lactarius peckia is chosen because it's a mycorrhizal species, and so it can become, okay, we need forest health to be able to even find our state mushroom.
Oh, yeah. Sorry, but could you define that word for us again? You said it was what type of?
Mycorrhizal, a mycorrhizal species, which means that the mushroom is growing in association with the tree roots in a beneficial symbiosis. I think I mentioned it very briefly at the beginning, but this is it's the same mushroom that needs the roots in order to live. It's trading nutrients like phosphorus, nitrogen for the sugars from the trees.
Gotcha. Okay, and I just noticed the time. I'm sorry, we are at 3, so I'm going to close this out. But this has been wonderful. I learned so much. And I think a really great way to close out too is to something you mentioned before, but there was a quote that was on the website that I was just enamored by, or by the report, actually, the 2023 report of the most valuable thing an individual can do is to diligently observe a small area year after year, or more often if you want, building a true picture of fungal diversity. And so since you mentioned very similar thoughts, I just wanted to put it in a end statement of if you're invested now that you've heard all this knowledge about the fungi around the world and around the continent of North America, especially, then that is a good thing to keep in mind. Of getting started is really not that challenging. And I will also close this out too on this page, just as a reminder to anyone who is watching of how to get involved. A good way to go is through SciStarter, so you can find the project and some basic details before you get started, and you can see how well you do over time. And I just want to thank you so much for joining us, Gabriella, for sharing all your wealth of knowledge, even though you're coming out of a cold. So thank you for taking the time to still be with us anyway. So thank you so much. I appreciate your time and all of the work you're doing with Fundus too. It looks amazing. It's going to be getting bigger and bigger over time, right? I'm glad that California was the starting point for states being involved. I hope there's many more that follow.
Yeah, thank you everybody for participating, for helping with this, for these podcasts in general. And thanks everybody for watching. We hope to see you get involved, join your mushroom club, and go out, learn mushrooms. They want to learn about you.
They want to learn about you? That's kind of ominous.
They're watching us too. They're looking at human diversity.
I think so. You never know.
Be surprised. They're more related to us than plants, right?
They are. Yep.
Yeah. Any last thoughts, Bob, before I
No, that sounds great. Thanks so much.
Awesome. Okay, for anyone who is listening, I'm just going to share my screen one more time in case you wanted to see the upcoming events. We have bats heading into spooky season with October. We're talking about bats on next week on Tuesday. Then we'll be talking about Redshift Wrangler, which is a NASA project in which we will be wrangling the cosmos through colors. We're using the colors used in or Redshift color. I'm probably describing this wrong. They're going to correct me in 2 weeks. And then we'll be talking to iWire, which is all about the brain and vision. So our ability to have vision based on our neural connections between the eyes and the brain. So really cool, exciting things coming up, and we cannot wait to bring it to you later then. And if you need any help, we have a million resources. So this is where I will close this out and say, email us if you need it. I will send a follow-up email to all of you who registered for all the resources we talked about too, because there were quite a few. I want to make sure I include everything for resources, including the website and everything there too. So thank you everyone for joining us. And I'm just double checking to see that our Zoom doesn't have anything question-wise out of there, but otherwise I think we're good to close. So thanks everyone for joining us and for listening to us later on. And we will be back next week and every week.